Hello A&P2 students at Madison Area Technical College. Today we started the reproductive system, and many of you joined online, but I failed to record that session, so I am recording a lecture and uploading it to YouTube for those of you that missed it, as well as those of you that want to review what we did today during class. So this lecture will involve an introduction to human reproduction. Most of it is review for you because we've already talked about sexual reproduction and meiosis in lab, and then we'll focus more specifically on just the male reproductive system. The topics on the details are included in study questions 19, 20, and 21. And you can check your textbook, chapter 27, for further reading. So, as you know, because you're human, the function of the reproductive system is to produce offspring by a certain method, sexual reproduction, as opposed to the way that bacteria or sponges reproduce. We actually form gametes, and those gametes have to join. So the gametes that are produced in the male reproductive system are called sperm. And another function of the male reproductive system is to introduce or deliver that sperm to the female reproductive tract. And within the female reproductive system, the gamete that's produced is called an ovum, and we produce several ova throughout our lifetimes. And the female reproductive system needs to deliver the ovum from the ovary to the uterus to meet up with the sperm. In addition, the female reproductive system needs to provide an environment that's nutrient rich for development of the embryo. And then it needs to kind of alter course and be able to make that environment a little bit less inhabitable and give birth to a fetus. So I've used this term sexual reproduction already, and I just want to use this pictorial to explain what I mean when I say sexual reproduction. I don't really mean anything in reference to the process of fertilization. Instead, I'm referring to the fact that we have cells in the ovary and the testis that are diploid, meaning they have two full sets of chromosomes. We refer to that or symbolize it with 2N. N being the number of different chromosomes, and we have 23 different chromosomes. From these diploid cells, we make gametes, and the process is called gametogenesis, or more specifically, in the female you could call it oogenesis, and in males you could call it spermatogenesis. The gametes that are produced are single cells, an ovum or a sperm, and each only has one set of chromosomes. So we refer to these cells as haploid. So instead of having a total of 46 chromosomes, there's only a total of 23. Homologous chromosomes have been separated from their partners. Then the process of fertilization begins, and the ovum and the sperm unite in fertilization to form a single cell that is now restored to the diploid condition, because two haploid cells coming together would make a diploid cell, and that diploid cell is called a zygote. So we were all just one cell at one time, a long time ago, and we were all zygotes. Now, how we become multicellular organisms is through the process of mitosis. So we grow, tissues, organs, all grow by the process of mitosis. And then at puberty, we are able to make gametes ourselves, and the process continues. Now, during the process of fertilization, the gametes coming together is called syngamy, and it brings the sex chromosomes together. And the sex chromosomes are the 23rd pair of chromosomes. This is a karyotype in which all the chromosomes are laid out so that you can look at any abnormalities. And the 22 pairs that aren't sex chromosomes are called autosomal chromosomes. The sex chromosomes include either, well, X and Y, and if X and Y are paired together, you get a male embryo. That's what develops. If instead two X chromosomes come together, one from the sperm and one from the oocyte, then you get a female. So notice that the only type of sex chromosome that a female can donate is an X, whereas a male can donate either an X or a Y to the gamete. So really it's males that determine the gender of offspring. So now we'll talk a little bit more specifically about the male reproductive system. So here's a sagittal section kind of showing the bladder and the reproductive system. You can even see part of the rectum here. The male reproductive system includes two types of organs: primary sex organs, also known as the testes. Their function is to produce gametes. And then the second type are just collectively called accessory reproductive organs. These include the structures of the external genitalia. That would be something that's not internal, something you could see visually on the outside. That would be the penis and the scrotum, where the testes are located. You can't actually see the testes though, because they're covered with skin and connective tissue. Other accessory reproductive organs include glands and then ducts. And the functions collectively of all these accessory reproductive organs are on keeping the sperm healthy and delivering it to the female reproductive system during fertilization processes. So we're going to look at each one of these organs individually, the anatomy and the function. We'll start with the primary sex organ, the testis. And the testis is covered by three membranes. The one that's most superficial and the one in the middle, both of those are derived from the peritoneum. So the peritoneum, you might remember, is the membrane that lines the abdominal cavity. And testes develop in the abdominal cavity. I'm going to go back a slide and show you where they develop roughly. They roughly develop up here. And they descend through the body wall during development, so that the testes actually pull down the peritoneum as they move outside the body. So that's how we get these two layers. And the name that's given to these two layers is tunica vaginalis. The innermost layer appears white on this picture, and that layer is called the tunica albuginea, and it's basically fibrous connective tissue. The tunica albuginea has partitions or septa that divide the testes into lobules. So these septa move inward and kind of cut the testis into individual sections, and these sections are called lobules. The lobules contain four ducts and the ducts are called seminiferous tubules. So in each lobule you'll have 4 seminiferous tubules. The number's not that important, I shouldn't have even mentioned it. But the seminiferous tubules is where sperm production occurs. So the sperm begin developing in the seminiferous tubules. And then as they develop and mature, they're going to move out into the reedy testes, then out an efferent ductule to what's called the epididymis. So the epididymis is this network of bunched up tubule that contains a head, kind of a mid-piece here, and a tail. And by the time the sperm get down to the tail of the epididymis, they're pretty much mature. This is really the site where they end their maturation, and they are stored until ejaculation. But when it's time excuse me, to deliver the sperm to the female reproductive system during ejaculation, then the sperm will move, and I'll use a different color here just so it's kind of clear. Then after ejaculation, the sperm is going to move from the epididymis through the ductus deferens. I often call it the vas deferens. That's what I learned when I was in school. And this vas deferens or ductus deferens becomes part of the spermatic cord, and it's going to move into the body and then make its way out the urethra. Okay, so that's where the sperm will end up, and we'll get there eventually. Right now we're just talking about the testes. So that's the overall anatomy of the testes, and what we really want to spend some time on is this structure of a lobule, or the seminiferous tubules in particular. So this picture right here shows you that there are regions of, I'm trying to figure out the best color for this. Probably white, actually. Let me find one. Okay, so this picture down here shows you seminiferous tubules. Here's one that's been cut, cross-sectioned. And you can even see it's been color-coded with a computer. Okay, your sperm are not blue. But you can see that the sperm with their flagella are developing interiorly. So this is the site of spermatogenesis. Now in between the seminiferous tubules you can also see some structures. There are blood vessels here, but there are also cells that are called interstitial cells, or cells of Leydig. Somebody named Leydig actually discovered them, I guess. And that's where testosterone is produced. Now the interesting thing about this is that testosterone is required for spermatogenesis. Required for sperm production is what I'm trying to write. So if the testosterone isn't even in the seminiferous tubules, if it's out here, how does it get in? Well, it turns out that there's some protein that's inside the seminiferous tubules that will actually draw the testosterone inward and bind to it so that it stays local. Some of the testosterone, of course, will enter blood vessels and travel throughout the body. And that testosterone is needed for the development of secondary sex characteristics, such as, hair growth on the chest, muscular growth, even skeletal growth, deep voice, all of the characteristics that we think of as male versus female, all of those are due to testosterone. Now there's one other kind of cell that I have listed here that's located in the seminiferous tubules, and these are the sustenocytes. I learned them as Sertoli cells, again named after a person probably. Also, they're sometimes called nurse cells. Just kind of funny because many of you are studying to be nurses, but what these nurse cells do is ensure that the sperm that are producing as well the cells that are producing sperm as well as the sperm that are produced stay healthy. So the Sertoli cells nurture spermatogenesis. Oh, I wanted to talk about this picture. So this is a more realistic picture that you would see after sectioning the testis and then staining with a typical, we call it an H&E stain, that gives kind of this purple pink appearance. And you can see a seminiferous tubule. I bracketed it. And you can see some blood vessels. Here's a blood vessel, here's a blood vessel. But you also see these groups in between seminiferous tubules. And these groups of cells are the interstitial cells, so the cells of Leydig. And that's where testosterone is produced. Inside the seminiferous tubules is a lumen, or a space, and that's where the sperm are going to flow through the tubule network. And within the wall of the seminiferous tubule you can see various cells. It's hard to really make out at this magnification the difference between different cell types, but I can tell right here that this cell is undergoing some sort of cell division because I can see some chromosome fragments or something. So we're going to see that sperm develop from the peripheral part of the seminiferous tubules and then move inward as they mature. So in here are germline cells that will actually become sperm over time. Another type of cell are the Sertoli cells, but I can't make them out. They're pretty big usually, so it's possible that this is one right here. And then surrounding the whole seminiferous tubule are cells called myoid cells. They're basically smooth muscle, like surrounding a blood vessel, and they can rhythmically contract to help the fluid that's in the lumen move, and that will carry the sperm throughout the network of the tubules. So in summary, the testis has many cells. It has germline cells that we would call spermatogonia. The spermatogonia actually develop into spermatocytes before meiosis happens. We will discuss that further. If that didn't make sense to you, don't worry about it. Just think of the seminiferous tubules as having germline cells that perform spermatogenesis. They also have cystinocytes or Sertoli cells that secrete growth factors, nutrients, and this androgen binding protein. That's the protein here that's in the seminiferous tubule that causes testosterone to stay localized. And the interstitial cells are the cells that make testosterone that I circled over here. Now, when testosterone acts locally like this, we call it a paracrine signal. And instead, if the testosterone enters a blood vessel and travels throughout the body, then we would say that's a hormone or an endocrine signal. So this slide gives you kind of a pictorial of what I was trying to draw. It shows you that germline cells are within this wall of the seminiferous tubule, as are Sertoli cells. They're in the wall. And the Sertoli cells make androgen binding protein, whereas the cells of Leydig, or the interstitial cells, I should really have that green, I'm sorry, I'll make it white at least. These cells make the testosterone, and so the testosterone is going to diffuse away from the cells in all directions that make it in all directions, but the androgen binding protein will keep some of it in the seminiferous tubules to promote spermatogenesis. To promote spermatogenesis. Okay, that's most of the testis. The rest of the function of the testis that I want to talk about has to do with the epididymis. So, as I mentioned, the epididymis is where, I don't have a picture here. That's crazy. I need to have a picture. The epididymis includes this head of the epididymis, and it also includes this tail of the epididymis. So it's quite a big structure. It's noticeable on the posterior side of the testis, and it collects all the mature sperm from the reedy testis. So what is the function of the epididymis besides just collecting all the mature sperm? The other function is to densely pack the sperm by reabsorbing some excess fluid, because fluid is constantly being produced excuse me, by Sertoli cells as well as those Leydig cells or interstitial cells. Fluid's always being made, and some of it has to be reabsorbed. So this is the site, the epididymis, where mature sperm are stored. They're densely packed, and they stay there until ejaculation or until they degenerate, because the lifetime or the lifespan of sperm is only like up to 2 months, so 40 to 60 days. After ejaculation, once the sperm enters the vagina, the sperm can live for up to 6 days. Okay, another accessory organ that we need to talk about is the scrotum. The scrotum basically protects and holds the testicles outside of the body cavity. That's its main function. Now, why does it do that? Why do we want skin out? Well, first of all, protection makes sense, but why are the testes outside the body? It's because our body temperature is 37 degrees Celsius. So our body cavity is at least that. And sperm production requires a cooler temperature, 34 to 35 degrees Celsius. So the testes need to be located outside of the body cavity. And of course we need something to hold them there, and that's the scrotum, composed of skin, fascia, muscle, blood vessels, and there's nervous tissue in there too, because there'll be nerves. But the scrotum, in addition to just kind of being a holding structure, also helps regulate the temperature of the testis. And it does that in a couple of ways. One is through this muscle called the cremaster muscle that I've kind of pointed to down here, but the cremaster muscle extends even farther up towards the inguinal canal, which is right there. So when this cremaster muscle contracts, that elevates the scrotum and therefore elevates the testis, and that increases the temperature of the testis. So this would only happen when the ambient temperature or the outside temperature is very low and the testicles get below 34 to 35 degrees Celsius. By bringing them closer to the body wall, the temperature will increase and sperm production can continue. Well, another problem is not getting too cold, but maybe getting too warm. So it's possible that the testis will need to be cooled at some point. And one thing that does that is simply by relaxing the cremaster muscle. But another thing has to do with blood flow. And the structure that's going to be important here is called the pampiniform plexus. That is a mouthful, huh? This is a network of veins that surround the testicular artery, or I should say the artery in the spermatic cord. So arterial blood is very warm. So here's the artery, and you can see it underneath, kind of peeking out between the pampiniform plexus veins. But arterial blood is warm. You know, it's going to come to the testis 37 to 39 degrees Celsius. Well, that's too warm for the testis. So what happens is the veins surrounding that artery can actually absorb the heat that radiates out of the blood. So if heat radiates out, then the venous, sorry, I better write, heat radiates out of the blood. The venous structure will carry the blood away from the testes. Therefore, the blood will be cooler by the time it gets down to the testes. So the pampiniform plexus, by being three-dimensionally 360 degrees around this artery, helps cool the arterial blood before it gets down into the scrotum to the testes, and it helps keep the testis temperature lower than our core body temperature. Another structure that's associated with the scrotum and the testes, as well as the spermatic cord, is the inguinal canal. So the inguinal canal is a passageway through the body wall between the scrotum, which is down here, and the body cavity, and it contains the spermatic cord. And the spermatic cord, in its simplest sense, I guess, would just be this ductus deferens, which contains the sperm. But, you know, the spermatic cord also, of course, has arterial blood supply going to the testes. It has venous blood returning from the testes. And, of course, we just talked about the pampiniform plexus. And there are nerves present in the spermatic cord that end up surrounding the testicle. And there's a layer of fascia in addition to the cremaster muscle. So the inguinal canal actually has quite a bit because the spermatic cord runs through there. Now, one condition that can happen is called herniation. And this happens because a location where the inguinal canal is kind of a weak spot in the body wall. So testes, as I mentioned previously, develop up in the body cavity in an embryo, and then over the course of development descend through the inguinal canal and come to reside in the scrotal sac outside the body. Well, this penetration through the body wall right there is kind of a weak spot, and there's tissue that covers the opening in many different directions, fascia, but it's still a weak spot. And one thing that can happen is that the small intestine can kind of puncture that. Now, this only happens usually with strain, some sort of physical strain, but the movement of the small intestine through this inguinal canal is called a hernia. In fact, any organ that moves to a location it's not supposed to be through some sort of membrane is a herniation. In this case, we would call this an inguinal hernia, because the herniation of the small intestine happens through the inguinal region. Okay, another structure for the male reproductive system is the penis. And the penis, as you probably know, is an organ that's designed to deliver sperm to the female reproductive tract, and therefore it has some structures inside that are vascular spaces that fill with blood to keep the penis rigid when needed. And these vascular spaces are called erectile bodies, generically. When the penis undergoes an erection, these spaces fill with blood, and the blood comes from the circulatory system. So you can see here is a blood vessel, here's a blood vessel, here's a blood vessel right there. So these vascular spaces are located in the shaft of the penis, as well as the head of the penis, or the glans penis. One is called the spongy urethra. I'm sorry, not called the spongy urethra, sorry. It's called the corpus spongiosum that surrounds the urethra. I made a mistake. This is the urethra right here. This is not a blood vessel. Please forgive me. And the other two are called corpora cavernosa. And when the blood vessels dilate, here are the blood vessels. There's some blood vessels there. When they dilate, blood moves out into these spaces. The reason they dilate is because the parasympathetic nervous system stimulates the wall of the vessels to release nitric oxide. And nitric oxide acts locally on the endothelium, causing vasodilation. And so these vascular spaces, or erectile bodies, the corpus spongiosum and the corpora cavernosa, they fill with blood, and that makes the penis erect and stiff, enabling it to penetrate into the vagina. Okay, let's look at some other organs, accessory organs of the male reproductive system, including the glands and ducts that help assist in delivering sperm, keeping it healthy. So we'll look at the function of these different things over time here. Let me first orient you to where we are in terms of the urinary system. I'm going to start there. So I'm going to do the urinary system in yellow. That's pretty appropriate. So this is a ureter, okay, and it's coming down from the kidney, and it travels behind the bladder and enters the bladder. And so here is where urine is located. So there's urine. When urine leaves the bladder, it has to exit the body through the urethra. And now comes kind of a mixture, if you will, or a joining of the male reproductive system and the male urinary system. The urethra extends right here through the prostate gland, and this is called the prostatic urethra, and then through the body wall, and here this urethra is called the membranous urethra, and then finally into the penis. And of course it's quite elongated, and this part of the urethra is called the spongy urethra. Now we're going to talk about the reproductive system and how it kind of joins this part of the urinary system. So here's where mature sperm are located, in the epididymis. They travel within the ductus deferens that's located inside the spermatic cord into the body cavity, posterior to the bladder, and here the vas deferens, or ductus deferens, kind of widens, and there's some smooth muscle that pulsates a little bit, and it helps send the sperm through this ejaculatory duct, which is right here. Okay, so this region of the vas deferens that's enlarged is called the ampulla, and then the ejaculatory duct leads from the ampulla. Now the seminal vesicle secretes some fluid that joins the sperm in the ejaculatory duct, and you can see the seminal vesicle is located on the posterior surface of the bladder as well. So sperm and seminal vesicle fluid enter the prostate, and the prostate makes its own fluid it contributes certain components to semen, and so that material will enter at the prostatic urethra and then join the membranous urethra. At the very base of the penis are another set of glands bilaterally, the bulbourethral glands, and they contribute just a fraction of the fluid in semen, but they secrete some important materials, and we're going to look at what those do in just a second. So the semen will contain sperm that comes from the ductus deferens. It will contain fluid from the seminal vesicle, fluid from the prostate gland, and fluid and materials from the bulbourethral glands. So now we're going to look at what is in here in terms of the semen. What does the semen contain? So the amount of semen that's usually ejaculated is somewhere between 2 and 5 milliliters, but that small amount can contain millions, 150 million sperms per milliliter, so hundreds of millions of sperm. The pH of the semen, or the fluid that's surrounding this sperm, is basic or alkaline. And this is important because the urethra in the male is somewhat acidic due to the presence of, urine in the past. And also the female vagina is acidic. And so this alkalinity will help keep the pH of the sperm regulated. It won't be exposed to too much acid. So semen contains about 10% sperm, 70% seminal vesicle fluid, and then most of the rest of it is fluid from the prostate. The bulbourethral fluid contributes very little to the volume, but it contains an important component that can clear the mucus. Well, we'll come back to that in just a second. It lubricates the penis and it neutralizes the acidic urine. We'll come back to that in just a second. And the seminal vesicle fluid is usually yellow in color, and it's mainly about providing an energy source for the sperm so that they have enough energy to make ATP, have some sort of organic compound to make ATP from, and neutralize the acidity of the female reproductive tract. Prostatic secretions also provide a carbohydrate source or an organic nutrient. But in addition, the prostate secretes things called prostaglandins, molecules that are called prostaglandins, that help the sperm move in the female reproductive tract. And I'll talk more about that in just a second. So one other thing I wanted to mention while we're on the accessory glands of the male reproductive system is a condition called benign prostatic hyperplasia, BPH for short. And what that is enlargement of the prostate gland. It gets bigger, okay, and that's due to mitosis. This is usually, well, this is not cancerous. It's benign, not malignant. So it's not like cancer will spread somewhere else or anything like that. But it's a little bit uncomfortable for a couple of reasons, and this happens in older men. One reason is because it puts pressure on the bladder and it causes an urge to urinate, even if the bladder is not completely full. Another thing is with the growth of the prostate, sometimes this urethra becomes somewhat impinged, or at least it doesn't relax as well, and so you have the urge to go, but you can't. It'd be very frustrating. One of the ways that they test to see if the prostate is enlarged due to cancer or just due to this typical kind of benign condition, is that they'll sample some cells and look for something called prostate-specific antigen, or PSA. And there are certain normal levels and abnormal levels. I don't know what those levels are, but it helps them determine whether or not the growth of the prostate is a cancerous condition or just benign and an aging mechanism. Either way, there's a treatment, even for benign prostatic hyperplasia. Okay, so we see that the main functions of the semen are to, I'm going to go down here first, deliver sperm to the female reproductive tract. In addition, to keep the sperm motile, because they're going to move by this flagellum that you see here. It'll start waving back and forth and propelling the sperm in that direction. To keep the sperm motile, a pH of 7.5 is needed. However, the male urethra and the vagina in the female has a pH lower than that. So the semen having an alkaline pH will help balance and keep the pH that the sperm are exposed to 7.5. In addition, you may have noticed that there are mitochondria located in the sperm here. I guess you could call it maybe the neck. We don't call it that. We call it the mid-piece. But these are all individual mitochondria that make ATP, and the ATP helps to fuel the movement of the flagellum. To make ATP, of course, you're going to need some sort of organic source and energy source, and that's what the semen provides in the form of carbohydrate. And last but not least, how is the sperm going to move through the female tract with all the secretions that the female reproductive tract has? Well, or maybe not very smooth surface. The bulbourethral fluid is really important for lubricating the female tract. It helps the sperm continue their movement or motility. Now, initially, when the sperm are released into the vagina, it's helpful to have the sperm stuck to the wall of the vagina just a bit, not for long term, but just so that the sperm don't come out when the penis withdraws. So there is another compound that's in the semen called semenogelin, and that's a little bit sticky. So the sperm tend to adhere to the female reproductive tract, but then as soon as that breaks down, the bulbourethral fluid allows lubrication. Now, in addition, this sperm is going to have to move through, I'm going to draw this, is going to have to move through the female human reproductive tract. I'm going to draw that right now. So here's the vagina. This is the uterus. I know I'm not the best artist. And with a mouse, it's not that easy either. And here's the horns of the uterus. Okay. Now the ovaries live out here. And the egg comes this way. So ovaries are out here. The sperm in the vagina, you know, they're going to stick for a little while, and then they have to propel and get through the uterus. Now, I have not shown this, but the uterus has an opening called the cervix. That little opening there is called the cervix. It's kind of tight. It relaxes at certain times. It's just smooth muscle. But for the sperm to make it through there, it's important for these prostaglandins to thin the mucus that's located in that region. So the cervix contains mucus that protects the uterus from any bacteria that might come up through the vagina. But of course we want sperm to be able to get through. So the prostaglandins thin the mucus. Okay, that's most of the structure of the male reproductive tract. Now we're going to look at function and talk about the formation of sperm. Sperm are formed in two stages. The first stage is called spermatogenesis, and spermatids are made. And then the second stage is called spermiogenesis, and the spermatids mature into what we call sperm. Now, the formation of sperm begins at puberty, and it's due to hormones, and it continues throughout the lifetime of a male. As you know, it happens in the seminiferous tubules, but the final maturation can occur in the epididymis. And this is where sperm become what we call self-motile, and I'll explain that as I progress. Okay, so spermatogenesis, the first step, involves what I've always called a germline cell, which is a stem cell. It involves a germline cell becoming a spermatogonia, or spermatogonium, I should say. So this is really a stem cell, and it is diploid, and it has the potential to become a sperm, but right now it's undifferentiated and it's a stem cell. It can divide, and when it divides, I shouldn't have done that. I'm going to do it a different way. When it divides, one of the daughter cells will replenish the number of stem cells, but the other daughter cell is called a primary spermatocyte that can, and it's down here too, it's the same thing, that is also diploid, because mitosis is the only thing that happened. So a primary spermatocyte originates through mitosis and then differentiation, and it's diploid. This primary spermatocyte goes through the first meiotic division, meiosis 1, to produce secondary spermatocytes. And those daughter cells are haploid, which we learned in lab last week. And they're haploid because homologous chromosomes have lost their partner. These secondary spermatocytes undergo meiosis 2 to produce spermatids. And there's 4 products that are viable, and all of them are also haploid. So they're all haploid. This process takes place, as you know, in the seminiferous tubule. And this particular picture shows the outside or the peripheral part of the seminiferous tubule over here. And this is the lumen region of the seminiferous tubule. Within the seminiferous tubule are some Sertoli cells, and I'm going to outline a Sertoli cell. It's this yellow thing. They're quite big, and interspersed throughout the cytoplasm, kind of like the way axons are surrounded by myelin. The spermatogonia in the periphery begin the process of spermatogenesis. So we first get the primary spermatocyte, and then we get the secondary spermatocyte. And this process continues next to the Sertoli cells all the way to the lumen region of the seminiferous tubule. So there's a lot of sperm produced each day, 400 million, and the process takes somewhere between 9 and 10 weeks, something like that. Now when we get to this spermatid stage, the cells that are going to become sperm are closer to the lumen. And that's helpful because they need to go through their next phase, which is going to be spermiogenesis, to become what we notice or what we recognize as a sperm with a flagellum. Then the Sertoli cells are going to retract, they kind of contract a little bit, and that loosens up these sperm, and fluid is actually going to move them or dislodge them and cause the movement of the sperm. So traveling of sperm throughout the duct, the seminiferous tubules and the duct work of the testes is actually just due to the movement of fluid. That's the flagellum is not working yet. So the second process, making the sperm look like what we recognize a sperm to look like, spermiogenesis, involves basically 4 processes. The flagellum forms, of course. But in addition to the flagellum forming, the head of the sperm, I know that's really bad, the head of the sperm forms what's called an acrosomal vesicle, which contains enzymes that can penetrate the ovum. Another thing that happens is that the secondary spermatocyte over time loses cytoplasm. And all that really is retained as far as cytoplasm goes is just enough to hold the multiple mitochondria in this mid-piece region. So the process of spermiogenesis involves this secondary spermatocyte forming an acrosomal vesicle, which you see matures over time, and it's eventually just called the acrosome, but it contains enzymes that help penetrate the egg. And then in addition, the centrioles start making lots of microtubules, and those microtubules grow in length to form the flagellum. So that happens over time. And in addition, you'll notice that there are more mitochondria that are made over time, and the multiple organelles, and they line up close to the flagellum, and excess cytoplasm is expelled. So that gives us what we call the mature sperm, with a head, a midpiece, and the tail. So these spermatids that are formed actually move due to the fluid pressure created by the continual production of fluid that the Sertoli cells make. So the Sertoli cells are constantly secreting fluid in. Now the interstitial cells do too a little bit, but the fluid doesn't really get into the lumen as well. So the fluid enters the lumen of the seminiferous tubule and pushes against these tails, and the sperm are propelled during down the length of the seminiferous tubule. The flagellum isn't used until the sperm enters the female reproductive tract. Okay, there's one more topic that relates to formation of sperm and maturation of sperm in the male reproductive tract that's under hormonal control. So we want to talk about testicular function a little bit more. Now, it is true that the hypothalamus and the anterior pituitary aren't completely developed until puberty. Most of the brain isn't really even developed until we're 20 or so, but nonetheless, maybe 26, but nonetheless, development of the male reproductive system occurs in the embryo, and in part it does so in response to hormones that are produced. But that production, hormone production, stops at some point in early development and then starts again in puberty. And the hypothalamus specifically starts releasing at puberty what we know as gonadotropin releasing hormone. And that gonadotropin releasing hormone travels through the hypophyseal portal system, which is a network of vessels that connect, their veins, that connect one capillary bed to another capillary bed. And so the gonadotropic gonadotropin releasing hormone enters the capillaries in the first capillary bed, travels through the portal veins, and then exits at the second capillary bed to affect the anterior pituitary, which you see right here. Now the anterior pituitary responds to gonadotropin releasing hormone by making gonadotropins. And the two gonadotropins that the anterior pituitary makes are luteinizing hormone and follicle stimulating hormone. And those are going to affect the testis, or testes, both of them, not just one. So, as LH and FSH travel through the blood, they have different effects on the testicle. The luteinizing hormone stimulates the production of testosterone, and testosterone is going to cause spermatogenesis. So the testosterone that's produced will cause sperm production. It also will travel throughout the bloodstream and cause the further development of secondary sex characteristics. The follicle stimulating hormone that comes from the anterior pituitary causes the Sertoli cells, which are in the seminiferous tubules, to make androgen binding protein. And that will keep the testosterone local, or at least at a certain level, and not allow it all to go out into the systemic circulation. Now, testosterone's able to kind of regulate its own blood level through negative feedback mechanism. So testosterone circulating throughout the systemic circulation is monitored by the hypothalamus, and if it gets too high, it'll shut down production of GnRH, which will usually shut down production of testosterone, but not androgen binding protein, so that sperm production will continue. A minimal amount of testosterone will still be produced and stay local. So this picture, again, it's very similar to what you've seen already, is just a depiction of the importance of androgen binding protein in keeping testosterone localized so that spermatogenesis can happen and not all of the testosterone leaves and goes into the bloodstream. Okay, the last few slides here are just questions for you to answer, and I've included the answers both in the notes and in another slide afterwards and highlighted red. So I hope this helped your understanding of the male reproductive tract, and I will talk to you again on Thursday. Thank you.